Alrighty, it's 5 o'clock, so we're going to get started. Um, Hello, everyone, and welcome to Café Scientifique from all of us here at SFU's Faculty of Science. My name is Stevie Benish, and I am the Manager of Outreach and Engagement here in the Faculty of Science. If you're one of our Cafe regulars, welcome back. And if you're new, thank you for joining us for the first time. This is our last Cafe Sci of 2025, and we are very excited to be wrapping it up with such an amazing speaker. If you don't know about Café Scientifique, it is the Faculty of Science public seminar series where our presenters share their scientific research and its impact on our local communities and larger society. We typically host about 6 of these discussions a year, all virtual. They're also all recorded, so you can view them from our website if you miss a talk, or if you'd like to share this with friends or family afterwards, please do so. And we always love to hear from you and hope you stay in touch. There will be a post-event email, so feel free to reach out. And there'll also be a link if you'd like to join our mailing list. And there you'll receive updates on future events. So before we get started, I just want to start us off in a good place by acknowledging that I am privileged to be joining you tonight from the unceded and ancestral lands of the Musqueam, Squamish, Tsleil-Waututh, and Coquitlam peoples. We thank them for having cared for and being stewards of these lands and waters since long before our time. We look forward to working with them in partnership as we continue to build and grow our communities together. If you know whose territory you're currently on, I would like to invite you to take just a moment to reflect on this. And if you don't know whose land you're currently on, a great resource for more information is the website native-land.ca. And there you can familiarize yourself with the Indigenous peoples whose lands that you live and work on. So, before we get to the talk, just a couple details to go over. Um, Note that this is being recorded and will be shared broadly. There is a live transcript available for your convenience. And following the talk tonight, we will have a dedicated Q&A time, but please feel free to drop any clarifying questions you might have throughout the talk just in the chat. During the Q&A time, please use the raise hand function. So you'll be able to find that at the bottom bar of your Zoom menu under React. So if you raise your hand, then we'll be able to unmute you and you can ask your question. Or if you would prefer, you can always type questions in the chat and I can ask them. We do ask that if you don't have a display name, please rename yourself just so we know who you are. And we would also love to see all your wonderful faces. So please turn your cameras on if you're comfortable, and especially during the Q&A part, so it can be a little bit more interactive. Alrighty, so let's get to tonight's discussion, the reason we're all here. So I am so pleased to be able to introduce Dr. Anais Remili, who is a postdoctoral fellow in the Department of Biological Sciences. Dr. Remili's research focuses on how industrial contaminants and so-called forever chemicals impact the health of killer whales and other apex marine predators within both polar and temperate ecosystems. Her work looks at marine mammal feeding ecology, ecotoxicology, and contaminant accumulation, and multiple stressor interactions and cumulative effects. Tonight, she will not only be sharing her research with us, but also some practical steps that we can take to help protect these animals and the waters that they call home. Dr. Remili is also the founder and chief editor of Whale Scientists, which is a platform that amplifies the voices of early career researchers and helps them to share their science with the public, which is what we're here tonight to do. So without further ado, over to you, Anais.
Thank you so much for the nice introduction. Can you tell me if this is all good?
It looks great.
Sweet, wonderful. All right, so thank you so much for having me tonight. I'm really excited to tell you a little bit about the research that we do here at Simon Fraser University. And some of what I will present today was also performed during my PhD at McGill University. But it's all linked and we all collaborate with each other and some really cool studies that we can do on marine mammals across Canada and even outside of Canada. So let's get started. Um, there you go. So marine mammals, as we know, they're kind of present in British Columbia and in our coastal waters, offshore waters. They're kind of everywhere. They occupy various levels of the food web. They're really important to the well-being of our oceans. So here's a broad food web system where we have our phytoplankton at the very bottom of the food web, and then our zooplankton, which gets consumed by fish, which gets consumed by other big fish or small marine mammals like seals, porpoises, dolphins, etc. Um, some of these small schooling fish can also get consumed by baleen whales, and everything kind of ends up in the mouth of our top predators, like the killer whale, for example. So we have marine mammals at each step of the food web, and they serve different service. They offer different ecosystem services based on what they're doing in the food web and how they interact with other species. So we know that, for example, the top predators like killer whales, they have a sort of control over the species that they eat. So if the killer whales went missing, it would most likely create a proliferation of the lower levels of the food web. So the other species would start kind of growing like crazy, and it would throw the whole balance out of balance. It would throw the whole food web out of balance. But baleen whales have a little bit of a different function in our ecosystems, and they're actually the fertilizers of our oceans because they ingest enormous quantities and they reject it in the form of feces. This feces is really rich in nitrogen and will come to fertilize the phytoplankton, which is at the very base of the food web. So baleen whales act like fertilizers, and top predators like killer whales kind of act as the regulators of our food webs. So those are some of the roles that they play. So I was just kind of showing this to tell you just how important different marine mammals are in our oceans and in the well-being of our ecosystems. But unfortunately, whales, no matter their size or their species, face a lot of human threat. So here I've drawn a few of the human threats that our whales can face, kind of worldwide, and some of them are applicable to BC as well. We know that there are risks of collision. I put it here. Can you see my little pointer or not?
Yes, we can.
Okay, sweet. Because otherwise, if I'm showing you stuff and I don't point it, it's not gonna be great. But we know that collisions can happen, and if you're based in BC, we've had a few very sad collision events in the past few months involving humpback whales, for example. So those are some of the risks that they can face when they're confronted to humans. We know that noise pollution is a big one, especially with our southern residents um, that are facing some of these threats, and it can affect their ability to perform echolocation and to find prey and communicate. We know that bycatch can also affect a lot of marine mammals. They can get caught up in the fishing nets. We know that whale watching can also be a pressure, and in some countries we have some swim with activities that can also disrupt the life of these animals. We know that on top of that, baleen whales, for example, are at a particular risk of ingesting large amounts of plastic. Um, and other whales like beaked whales can ingest some plastic as well because it kind of resembles their prey. Um, and we know that, of course, and that's in the case of the southern residents, overfishing and the lack of prey can be a big risk because if the whales don't have enough to eat, that's an added stress to the whale population. But there is also another thread that's kind of silent and cannot be seen, and they're called forever chemicals in mass media, but we call them persistent organic pollutants. So those are a type. We're gonna see all about that. I'm gonna tell you all about these chemicals in the next few minutes. Well, in the rest of the talk, but yeah, forever chemicals are another stress to these whales, and they're another threat that may affect their health. And so we can look at all of these threats together, and it sort of creates this environment that becomes really stressful, of course, for the whales, and all of these add up and sort of come and play a role in the population's dynamic, and some of them can even cause issues with the recovery of an endangered population, like we see, for example, in the southern residents. But so I'm an ecotoxicologist. So my main job is to look at the relationship between the ecology, so where the whales eat, and the toxicology, so how the whales are affected by contaminants. And so we know that contaminants mainly enter the whales' bodies through their mouths. And we know that this can be a really big issue for killer whales because of a phenomenon called biomagnification. So I have here a little slide that explains a little bit of the concept of biomagnification. So let's say a contaminant, typically when we talk about these forever chemicals, these persistent organic pollutants, we're talking about a molecule, a chemical molecule that has a strong affinity to lipids. And so these molecules will travel in the ocean and in the atmosphere. They're also volatile. And they will enter a body of water where they will get picked up by plankton because plankton is typically really rich in fat. It's a lipid-rich creature. And so it will ingest some of these contaminants and they will stick to the fat inside of the plankton. So when the plankton gets eaten by a fish, let's say, the fish will ingest everything that's already in the water in its environment, but it will also ingest everything that's been accumulating inside the plankton. So you get a round of amplification in these chemicals. And so the fish gets more contaminated than the prey it's eaten. And so then when the fish gets eaten by, let's say, a seal, the seal will accumulate everything that's already in the environment, plus everything that's amplified through the plankton and through the fish. So you get another round of amplification. So you get um, a higher contamination in these seals. And then, of course, if the seal gets eaten by a killer whale, the killer whale will have accumulated all these rounds of amplification in these chemicals. And so they will have a lot of chemicals in their bodies. And we know that that can cause some issues. That's kind of why we're really interested in looking at these chemicals inside the whales. And so I just mentioned to you that these chemicals enter, and I promise, I will explain uh, what these chemicals are, how they were invented, etc., a little bit later in the talk. But first, of course, we know that they enter the body of the whale through the mouth, but we need to figure out what the whales are eating. So in the case of British Columbia, we're pretty lucky because we can observe some killer whales feeding pretty much all year round. But it's not necessarily the case in other populations. So I'll replay this video real quick. They're from our really good friends of the Norwegian Orca Survey. So this drone footage is documenting a feeding on the seal by a pod of killer whales up in Norway. So it's really valuable to get this type of information. And it tells us that this pod specifically has been feeding on a seal at this point in time. So we know that seals are probably included in their overall diet. But this type of event is extremely difficult to observe all year, especially in Norway and in other Nordic countries where it's pretty dark half of the year. So we can't really deploy a drone and have some good visibility of what's going on at the surface. It's also limited to surface events because whatever is going on up to 3 meters, maybe we can see, but below that, we won't be able to catch it with a drone. It's also really challenging to deploy a drone in remote areas that may also have some really bad weather, for example, in the Arctic. And it can be difficult to establish long-term diets based on observation only. There is a little bit of an exception to this rule when you have constant access to the whales and you're able to follow them year round because they're staying along the shore and you have some pretty good teams deployed like we do in British Columbia and in Washington State that can identify these individuals and report whatever's been eaten by these populations, which is extremely valuable. But unfortunately, that's not what's happening in most of the populations of killer whales worldwide. So there are a few things that we can do to try to identify what the whales eat. There's a bunch of different techniques, like, for example, observation. You can collect the feces and look at what they've eaten. But in the last few decades, ecologists like myself have started to rely on skin biopsies. So they look like this little piece of beige tissue in this tube, and they're basically the blubber, so the fat that's right under the skin. So we typically collect these. I'll show you in a few slides because I have a video throwing little darts at the whales that will come and take basically like a little skin core that looks about as big as the first phalanx of my pinky. Um, and so these can be extremely valuable because we can run so many different analyses on these two different tissues. So we have the skin and the blubber. So in the skin, we can measure chemicals such as mercury, so heavy metals. We can also measure something called stable isotopes, which I won't talk about in this talk, but is a really nice chemical tracer we call that helps us to understand where in the food web the predator has fed. And then we have the blubber, and in the blubber, we can look at different things as well. So we can look at fatty acids, which are essentially lipids. We can do other types of isotopes, and we can look at organic contaminants, so those like persistent organic pollutants that are in the blubber. They're also telling us a little bit about the trophic level, so the position in the food web of our predators, because the more there are of these contaminants, the higher in the food web our animals are. So all of these are kind of connected and tell us information about the diet of whales. But for the sake of the first part of the presentation, I'll talk about fatty acids. So fatty acids are lipids. They're found in abundance in marine mammals' adipose tissue, and that is because marine mammals have a lot of blubber. They need a lot of energy, and they need to stay warm in cold waters. So fatty acids come in all shapes and sizes, and based on their shape or size, it gives them different functions such as thermoregulation or energy storage. There's a nice trick about fatty acids that they don't vary much from the diet to the predator, at least those insaturated fatty acids. So those here, those two at the bottom, they sort of retain the information of the prey when they get deposited into the predator's blubber. So, for example, if a whale here has eaten a herring, it will integrate the fatty acid signature from the herring into its own blubber. But if it fed on a ring seal, it will integrate the fatty acid signature from the ring seal into its own blubber. And so we can use this concept and sort of give it a mathematical twist. And we can basically come up with a model that estimates the proportion of different prey species in the diet of killer whales. So it's called quantitative fatty acid signature analysis. It's a bit long. We call it QFASA for short. And it's a model that helps us to estimate the proportions of prey species that are most likely to account for the fatty acid composition inside the killer whales. So we just applied this concept with a little bit of fun math. So first we need the fatty acid signatures. So those are all the lipids in the killer whales and in their potential prey. Then we need something called calibration coefficients, and we calculated those using captive individuals from SeaWorld. And they help us to understand how the metabolism of the killer whales work because there is a tiny modification between the diet and the predator's blubber. It's not a perfect one-to-one ratio, so we actually relied on captive individuals to understand how killer whales accumulate fat from their diet. And then finally, we need a statistical model to estimate the proportion of each prey in the predator's diet. So before I show you the results of that study applied to North Atlantic killer whales, I'll take you behind the scenes with me to Iceland, where I spent some time during my PhD doing some field work with the Icelandic Orca project. So this was a video that I took during field work of killer whales. It was a beautiful day. We shot the little dart. I didn't want to spook anyone, but it basically is like a little dart that we throw at the whale. And once back in the lab at McGill, I deal with the biopsies. And so I'll put them in really strong chemicals, such as chloroform, for example, which you must have heard of because it's really popular in media. Chloroform, methanol, and other really strong solvents. And basically, I'll extract and purify all the fatty acids inside this little piece of fat. And then once I'll have all my little extracts, they're in those little vials. And I put them into a big machine called a gas chromatographer, which will basically show me little peaks of each fatty acids. A peak is an intensity of a signal, and basically we can convert that to percentages, and we can get a signature of all those different lipids inside the whale's blubber. So this is the fun, the fun part for me, but it's kind of dreadful to look at. It's a wall of data, and this is basically the outcome of all these little videos that I showed you. Once I get my data, it looks like this, and so I have to go play with it, which is my favorite part of doing research. I love. It doesn't look fun like this, but I swear it's fun. Um, we have to kind of put our detective hats on and work and try to understand, put it all in the models, refine the models, and try to understand what the whales eat. And this is the data that we can get. So this is some of the work again I did during my PhD, where we did the largest transatlantic study on killer whales. We had 200 individuals, and we managed to estimate the diet of each population of killer whales. And so what we found is that the killer whales in the east of the North Atlantic, so in Norway, the Faroe Islands, and Iceland, they seem to prefer eating fish with a strong dominance of herring here. There was a little bit of mackerel as well in the Faroe Islands. And there was some marine mammal consumption as well in Iceland, Norway, and the Faroe Islands. Then when we went to the Greenlandic population, we found that they feed mostly on seals. So those are all the blue bars. So harp and hooded seal were the dominant prey. And then we had a little bit of mackerel consumption as well. And we know that there is lots of mackerel along the coast of Greenland, so it sort of made sense to us. Um, then in the Canadian Arctic, we had a consumption of narwhals and belugas, which seemed to match some of the reports from the studies out there, as well as ring seals, which seem to be an important prey as well. In eastern Canada, the dominant prey were baleen whales, which also seems to match some of the observations out there, as well as harbor porpoise and a little bit of seal. So this was really neat because it allowed us to do this really big transatlantic study and look at different populations, but we can actually dive deeper and look at the diet of each whale inside a population. So I'll only show you 3 examples for the sake of time. But we can observe some patterns inside the populations. So, for example, in the Canadian Arctic, we found that about half of the individuals, so each vertical bar here on this graph is an individual and the composition of their diet. So, for example, this individual here had a diet that was exclusively belugas and narwhals, according to fatty acids, versus this one, which was exclusively ring seal. And so in the Canadian Arctic, we had sort of like a two-way specialization, either on cetaceans, so belugas and narwhals, or on pinnipeds, such as ring seals. So that was quite interesting to see that we had sort of an interesting pattern out there in the Canadian Arctic. But when we moved to the Greenlandic population, we observed a completely different pattern. This pattern shows us that each whale kind of eats a little bit of everything, and we don't really have a clear preference. We seem to have a dominance of this harp and hooded seal here category, but the whales seem to feed on pretty much whatever they can find. So we think they're being opportunistic predators out there, and they're just kind of putting their teeth into whatever they can find. But then when we go to Iceland, it's something completely different where we get a specialization on herring here, which seems to dominate most of the diet of the individuals in Iceland. We seem to have a gradient of predation on marine mammals, though, and that seems to match what we've observed in Iceland. We know that there's a group of the Icelandic killer whales that like to migrate to Scotland. And when they go to Scotland, we know that they target seals and other marine mammals. We have also had observations of killer whales in Iceland eating porpoises. And so it seems that there are some individuals that like to switch prey, and we can measure that. We can detect it using just the fat in their blubber. So that's actually pretty cool, and it allows us to have a little window into the lives of these individuals without having the need to observe them year long. Because it is pretty hard to go to Greenland or the Canadian Arctic and observe these whales all year and obtain these beautiful drone footage, like I showed previously with the Norwegian guys. So each of these studies, each of these chemical tracer that we call them studies, is a piece of the puzzle basically that helps us to understand what the whales eat. Um, and I know it's like crazy to think about, but there really are people that are extremely interested in what killer whales eat, and they're going to combine all of these methods to really solve this puzzle and try to understand because it has such important consequences, not only for their contaminant levels, but also for their role in the food web, like I showed in the first slide. So it's it's also a nice detective work, and you kind of get addicted to it when you get started, and you really want to know like what the whales are eating. Are the studies agreeing with each other? Or are they disagreeing? How can we solve the mystery? It's really fun, and we could do that using a biopsy, which is great. But we can use other methods as well to confirm what these indirect methods, such as feeding tracers. So, like for example, the lipids, they're indirect methods, and we can use other methods to confirm what's been estimated by the models. And so we can use photo identification. So this is what we do a lot here in British Columbia with our resident and transient killer whales. We can look at the stomach contents in deceased individuals, so stranded animals. That can be a hit or miss because sometimes the animal is sick and has an empty stomach. But sometimes, if it's a blunt force trauma and the animal ends up dying, we can look at the stomach and see if there are any hard parts that we can identify, and that gives us an idea of what the whales eat. And we can also collect the feces. So that's something that we're really good at in British Columbia and in Washington State, where we can collect the feces of various resident killer whale populations, for example, and scan the DNA that's present in the feces to try to understand what the whales have been feeding on. So these days, I can't show you the results because we haven't published the paper yet, but we just applied this fatty acid method to the southern resident killer whales. And so keep an eye out for that paper because it's supposed to be submitted very soon, hopefully in the next month. And yeah, we're finding some pretty cool results and trying to assemble the pieces of the puzzle to understand the diet of southern resident killer whales. So, knowing the diet is really important. I'm sure I've convinced you of that. And in my work as an ecotoxicologist, it's extremely important to know what the whales are eating because it's going to influence how much contaminant they accumulate. And so, coming back to our killer whale, we care a lot about contaminants in killer whales because, well, they have a lot of fats. They accumulate a lot of these contaminants. They also have a really long lifespan, um, almost as much as humans, and so they can accumulate these contaminants throughout their entire life. We also know that because of biomagnification, which is the slide I showed you in the very beginning about the amplification of these chemicals through the food web, they can accumulate crazy amounts of contaminants. And on top of that, killer whales, just like other cetaceans, are pretty unlucky because they have an inactive gene in their DNA that was proven in 2018. And this gene helps other species to detox their bodies when they accumulate these contaminants. But unfortunately, cetaceans don't have that gene active, and so they can't detox their bodies when they start accumulating these contaminants. So as a result, killer whales are the most contaminated animals on the planet. Um, but when we talk about these contaminants, maybe I should give you a little bit of background on what contaminants exactly we're talking about, because you heard forever chemicals and you heard persistent organic pollutants. So I'm going to tell you a little bit about where they came from and what they were used for. So these contaminants came, there's different types of contaminants. We have the old ones, the old guys called the legacy contaminants, and they include about 12, they included 12 contaminants at the very beginning of when we started to really freak out about those. And they're called the dirty dozen. And that is because they're extremely persistent and extremely bad for the environment. So unfortunately, like many stories that ended badly, it started with Monsanto, and Monsanto developed PCBs, so polychlorinated biphenyls, in the 30s, and they were chemicals that had a really there were basically two phenyl groups. Stuck together that had chlorine atoms around the structure. So they were extremely stable, which was their main property. We used these chemicals because they were extremely stable. And so they would stabilize conductors, they would stabilize paint, they would stabilize plastics. So we started using them everywhere. And they were great at what they did because they really helped us develop our industries. Um, and there's another class of another type of chemicals in this dirty dozen, so in these old chemicals. One of them is called DDT. It was called neoside back in the days. And we know about DDT, and you might have heard of it because it was extremely popular in the 1970s. It almost caused the disappearance of the bald eagles in the United States because it was causing the eggshells to to become so thin that they would crack, and basically the eagles could not reproduce. So DDT was really bad, and we started banning it in the 70s. Chlordin is another one that was invented around the same time, and both DDT and chlordins were pesticides. And so we used them on our crops because they were extremely good at eradicating all the pests. Unfortunately, they eradicated a lot more things than just the pest. And that is because these chemicals can end up into the environment. So what will happen is that the PCBs, for example, that were manufactured ended up entering our atmosphere or a hydrosphere through the burning or through the reactions. They started to enter the atmosphere where they would circulate freely until they reached the poles where they would precipitate in the form of rain and snow. Or they would just leach out into the rivers and start entering the environment that way. And pesticides were just sprayed on our crops, and so they would leach and enter our environments through river after every rain event, where they would slowly travel down and end up back into the lakes and oceans. And so that created a big issue, but that issue was silent for a long time. So as I mentioned, we invented these in the 30s, and we started to really use them massively all through the 50s and 60s. And we had no idea what was going on because we can't see these chemicals; they're invisible. We can't smell them; they don't have a smell. We can't really taste them, and we can't hear them. So they were just an invisible menace um, and silent killers until the 60s, when a few Swedish researchers were conducting water sampling in a lake. And in the lake, there were a lot of fish that were dying. And so they ran the water sample through a gas chromatographer, which is the same machine that we analyze fatty acids on. And they started to see these huge peaks. And they found out these peaks were actually those polychlorinated biphenyls that were used in the industry and many miles away, hundreds of miles away, to create these plastics, these paints, transformers, conductors, etc. And so that's when people and scientists started to realize that all of these chemicals eventually ended up into our environments and into our watersheds. So that created a little bit of a panic movement through the quote unquote developed countries, where countries started to ban contaminants. And in 2001, the United Nations formed the Stockholm Convention, which lists and bans all of these chemicals. And so in 2001, the Nations voted on banning these dirty dozen, so all the old chemicals, which included those polychlorinated biphenyls, so PCBs, as well as the DDT and chlordins, which are part of those pesticides. So we started to ban those effective in 2004, and we started to add other chemicals to the list. What I want you to realize is that so we have 12 chemicals initially listed on the United Nations. Today in the world, we have over 350,000 chemicals in use. Most of them, we have no idea what they do, and most of them are just being used without any reported toxicity threshold or anything like that. So we're focusing on these 12 because they are incredibly destructive and incredibly high in our environments. But this is only the very, very tip of the iceberg. And so the Stockholm Convention started to list these 12 initial contaminants and started to ban them. And so what did we do? We replaced them with other chemicals. So because these chemicals became illegal, they got banned, the industry still needed something to add to, for example, our couches so they wouldn't catch on fire. So we took basically more or less the structure of a PCB, but we just replaced the chlorine atoms by bromine atoms. And we came with PBDEs. And those were great. And they slowed down the burning in our couches. We started using them in firefighter foam, etc., fire extinguisher. And they worked really well until the same story happened and we realized that PBDEs accumulate in the environment. And so today they have been added to the Stockholm Convention. And we trace them in killer whales and we measure high levels of them in killer whales. So then we were like, okay, let's calm down on the PBDEs. We're going to ban them. So the industry came back with PFAS, the new forever chemical. And so PFAS can be found in your Teflon cooking pans or in your water repellent coatings in your jackets. And again, they're great. They work really well in their intended use. So they really work well in a Teflon pan, and your Arc'teryx jacket really is good at repelling the water. But slowly, these chemicals are leaching into the environment through little particles, and they go and they accumulate in marine life, and they threaten their health. So it's a little bit of a vicious cycle that we keep repeating, where we ban things and then replace them with something else without proper testing, and then decades later, we find out that these products are also not good for the environment. So if we come back to our killer whales, the neat part about our design in the transatlantic study is that basically when we collected those little biopsies, we separated the killer whale blubber into two equal parts. The first part was used for the fatty acids to help us understand what the whales were eating, which was the graphs that I showed you earlier. And the second half was saved to measure the persistent organic pollutants inside the whales. So those included the dirty dozen, so the PCBs, all these pesticides. So we have DDTs, chlordins, those are the two most prevalent ones. We also have HCH, chlorobenzene, etc. Those were all used to control pests at some point in time. And they were very chlorinated, very persistent in the environment. And then we have those flame retardants, so the PBDEs that I mentioned, we measured them, and we also focused on new chemicals that have replaced PBDEs essentially, that we can also detect inside the whales. And so we know that these chemicals altogether can have some effects on the animals that can be quite traumatic. We know that past a certain point, PCBs can start to weaken the immune system of marine mammals. We know that when they get really high, they can start to disrupt the hormones inside the animals. They can even lead to reproductive failure which was measured in seals in the 1960s, where they found that literally the mother's uterus was showing some occlusions, so the mother could not carry a fetus inside her womb. We know that they've been linked to miscarriage in some marine mammals, and we know that some of these contaminants also caused intestinal cancer in belugas in the St. Lawrence River. So we know that they can do quite a lot of damage, but we had no idea what the concentrations were across the North Atlantic, especially for the western part of the North Atlantic. We had no idea how high or low contaminants were in our Canadian killer whales, but on the other coast. So we measured all these contaminants, and this is the result. But I will take you step by step because this is a bit of a crazy graph if you're not used to it. So we found that the highest levels of contaminants across all these different classes, so PCBs, DDTs, chloridins, and the others, were the highest in the west side of the North Atlantic, so Canadian Arctic and Eastern Canada, and decreased a little bit in Greenland, and then decreased a bit in Iceland, and were the lowest in the Faroe Islands and Norway. So that was interesting because it goes against all the patterns of atmospheric and hydrospheric currents, where usually stuff produced in North America get pushed to Europe. So if you look at contaminants inside the snow, for example, or just the water, or very low trophic level animals such as plankton, the contaminants will typically be higher on the east part of the North Atlantic than on the west part. But we observed the opposite trend here in our killer whales. I will also draw here two lines because these concentrations probably mean nothing to you if you're not an ecotoxicologist, which makes total sense. So the first line here is the orange one, which shows the threshold for having risks of immune effects in marine mammals. So this is for the purple bars here, there are PCBs. That's the contaminants for which the threshold was calculated. So we see that for 4 of our 6 locations, the PCBs exceeded that orange threshold. And then the red bar here is the threshold for having risks of reproductive failure. So that was calculated for Baltic seals back in the 70s. And to give you a bit of a secret, of behind the scenes secret, that was something that we cannot do today, where we cannot give contaminated food to seals and then cut them up and see how it messed up their bodies, because it's extremely unethical. Back in the 70s, people did not have too much consideration for the seals in general, so they were allowed to do that. And they calculated that threshold. So it does not necessarily mean that all the killer whales are unable to reproduce. It just means that this is the highest threshold that we have for risks on marine mammals. And 4 of these populations are exceeding that highest threshold that we have for marine mammals. So this is not great news for North Atlantic killer whales, and it's really not great news for these guys on the west side of the North Atlantic, so on the Canada side of the North Atlantic. We also looked at differences inside the populations, and we tried to understand if there was an effect of sex or diet and which one has the strongest effect in our contaminant concentrations. We found a significant effect of sex when we saw that adult males had more contaminants than juveniles and females. So why is that? Well, we know that females, we expected that result because females are typically less contaminated than males. And that is because females need to produce milk for their babies to feed them, to make sure they grow healthy and quickly. But to do so, to produce this fat-rich milk, they need to use some of their own blubber. And by metabolizing the lipids inside their blubber, they will also pass down up to 70% of the contaminants that they have stored inside their blubber. So that can be quite a shock for a developing calf. And we know that it can most likely cause some developmental issues, maybe some immune issues where the calf cannot fight against diseases or parasites, maybe some endocrine issues where the calf cannot develop properly and maybe have some reproductive issues in the future. So we know that can cause a lot of problems. But that is basically why females are less contaminated than males. And then we also saw that there was a strong effect of diet. In fact, the strongest effect in our study was from the diet. And we know that the killer whales that fed on herring, and all these results came from the lipids and our lipid model that we applied to the same animals. We know that the killer whales that fed on fish were less contaminated, and those that ate a mixed diet of marine mammal and fish were in the middle. And then the most contaminated ones were the guys feeding on pinnipeds and toothed whales. Um, and so that makes sense because it all comes back to this biomagnification concept that I introduced at the beginning of the talk. But basically, what it tells us is that the killer whales feeding on these marine mammals on the west side of the North Atlantic are showing risks of having issues to their health because of the amounts of contaminants they're accumulating from their prey. So those were not excellent news for the killer whales, but it was definitely a motivator for us to really start diving into the health effects of animals. So before I jump onto some of the work that we did with health effects, um, I will just like do a little bit of a side conversation because I've received specifically from people on the West Coast, I've received a lot of questions in the past when I've introduced this concept and this study, which was wait, big killer whales are extremely contaminated, but they're not dying. In fact, they're doing really well, and we know that they're some of the most contaminated killer whales. Um, so to that, I'll say yes, absolutely, you're right. Killer whales, big killer whales seem to be doing okay. Um, I will hype up the upcoming study of one of my dear friends and colleagues, Chloe Kotek, who is working with DFO and with basontology on Big's killer whales using photo identification, and is showing that there is still a big transfer of contaminants happening to the calves, and that may impact some of the calves if the female hasn't had a calf in a long time because she's passing down so many contaminants that it may cause the death of the calf. So keep an eye out for that paper because it's going to be really big when it comes out. Um, but in the meantime, let's consider this big plump male, Big's male, who feeds on pinnipeds and other marine mammals, has a crazy high amount of contaminants. But for right now, in this big plump male, all the contaminants are being stored sort of safely inside the blubber of the animal, where they're away from the blood vessels and any circulation to the whale's organs. But if something happens to the animal, let's say old age or a disease or parasite or something like that, and the animal becomes thinner from losing some of its blubber because it needs energy, so it converts that blubber into energy. Some of the contaminants will get more concentrated inside the blubber because they're so lipophilic that they will stick to the fat no matter what, so they stay inside the blubber. But some of these contaminants will come with the lipid inside the blood vessels as the lipid gets transformed into energy. And so these chemicals, these PCBs, for example, will get down to the blood vessels where if the animal loses weight too quickly, they can have some acute toxic effects inside the animal's body. So they can come and target organs. They can have a whole cascade of different toxic reactions. And so that is a concern. And that is why, for example, we are always trying to push the message that killer whales need enough food. And of course, in the case of southern residents, that's an extreme concern because we know that not only they don't have enough food to survive, but if they become hungry, all the contaminants that they have inside their blubber some of it will get passed down their blood vessels and potentially come and stress the animal even more and cause more issues, which can lead eventually to a premature death. So bigs are okay for now, but keep an eye out because Chloe also has some incredible papers coming soon, where she'll look at the steroid hormones inside of big killer whales and link it to contaminants and try to see if there is a relationship between stress hormones like cortisol and contaminants. So that's Chloe Kotick from the University of Alaska Fairbanks, working with bay cytology and Fisheries and Oceans Canada, doing some incredible work on big killer whales and contaminants. Okay, I'm done fangirling. What does this mean for West Atlantic killer whales? Well, we know that those toxicity thresholds were exceeded in the west of the North Atlantic. But as I mentioned, those toxicity thresholds came from other animals, and at a time where it was okay to feed contaminants to an animal and see how its body reacted, which is absolutely not what we do these days. So we need new toxicity thresholds to be able to understand how these contaminants affect these animals. Still today, even though they've been banned for decades. So some of that could come from in vitro work with cell lines. For example, we have some colleagues, Pierre Blevin and his colleagues from Norway are doing some incredible work on killer whale blubber biopsies, where they literally take the thinnest little scalpel and cut the thinnest slice of tissue, and then they incubate it and they throw some contaminants on the tissue and see how it cascades and creates all these problems in the tissue, which is absolutely revolutionary because we can get an idea of how contaminants actually affect the tissue without dosing a whale, which obviously is not what we want to do. So really cool stuff that they're doing in Norway as well. We also know that contaminant cocktails are potentially more toxic than one single class of chemicals. So for example, if we look at just the toxic effects of PCBs, let's say we're going to dose a rat and put PCBs in its bodies. Well, we know that putting PCBs plus DDTs plus something else, even if they're all in the end at the same concentration, the contaminant cocktail will be more toxic than a single contaminant. So we know that it could create a lot of different effects inside the animals' bodies, but we don't exactly know what these effects are yet. So there are new techniques called omics, which I will introduce in the next few slides, that help us to understand how the metabolism of the animals is being impacted by contaminants. And we can do that in an indirect way, which is very nice because, again, we don't want to be dosing these animals. They're going through a lot and enough already. And then hormones is another way that we can determine the effect of these chemicals on the animals, because we know that hormones can be disrupted by these chemicals. And if we look at, for example, stress hormones like cortisol, we can try to understand if maybe there's an effect of these chemicals globally. So I'm going to tell you a little bit about omics, and I realize that this is becoming a little heavy, so I won't spend too much time on omics, but I'll introduce the concept a little bit and tell you about some of the work that we've been doing here at Simon Fraser in Professor Tanya Brown's lab on different Arctic marine predators. So not killer whales yet. We're working on killer whales, but the results aren't ready yet. But we've been doing a lot of really cool studies on polar bears, ring seals, and some of my colleagues at the lab are also doing some really cool work on belugas in the St. Lawrence estuary, which are endangered. So some really cool work coming out of omics. So before I tell you about omics, I'll just mention that in the north, the contaminants are not the only threat. We also, of course, have the threat of climate change, which may cause a loss of sea ice, disrupt the food web, cause some nutritional stress. And as I just mentioned two slides ago, nutritional stress can in turn have an effect on contaminants, which can cause some cascading effects and have some really bad health consequences for an animal. So that is an added stressor that we try to look at using omics. So omics and particularly metabolomics, which we are focusing on in the Brown Lab, are some really large scale studies measuring more than 600 different metabolites. So, metabolites are small molecules in organisms. They're used to reveal how the exposure to contaminants and other stressors disrupt the biochemical pathways and the overall health and metabolism of the animals. So we look at bile acids, we look at bioamine, we look at amino acids, we look at the fatty acid metabolism. We measure all these 600 plus metabolites, and basically we do a little bit of math magic to try to understand exactly how the metabolism is being impacted by different stressors. And so it's really cool because we need a tiny, tiny, tiny amount of skin or liver or blood or something else. These days, we're also looking at it in poo samples. We're looking at it in urine samples. We can really measure it on many different types of matrices. And it's also being used in human research today to look at, for example, the impact of several cancers, et cetera, et cetera. So metabolomics are being used really like worldwide everywhere in biology these days because they're really helpful at helping us understand the tiny little mechanisms that are being impacted by different stressors, whether it's a disease or contaminants. So we use that. I'll just show you the tiny recap because I don't want to dive too deep into it because it starts to get a little tricky. So we did that in polar bears in the Canadian Arctic, and we compared two different populations of polar bears and two different populations of ringed seals using these metabolomics assays. And we tried to understand if there was an effect of persistent organic pollutants on the health of the different populations. And what we found is that PFAS, so those forever chemicals that are in the Teflon pans or your waterproof jacket, actually had an impact on the lipid metabolism of animals, on the bile acid synthesis, on the inflammation signals inside their liver, and on their overall oxidative stress inside the liver. So there was more markers of oxidative stress linked to higher PFAS, and PFAS was really the one that had like the strongest effect on these animals' health. So it's kind of disheartening because it tells us that polar bears, even in the high Arctic, already are dealing with the effects of climate change, but also have to deal with the impact of contaminants that we're producing here in the south, really far away. And it's great for us to be able to prove it, but it's also a bit of sad news. But we use these metabolomic assays as well, using a previous assay that only measured 250 metabolites, but our new ones measure 600 plus. And we used it in two different tissues. We were able to show that in the liver, we had an impact of contaminants, so PCBs, DDTs, chloridins, et cetera, as well as mercury that was linked to oxidative stress again in those ring seals from those guys came from Labrador. And we showed some pathways disruption, the protein pathways. So we could measure contaminant effect in these seals. But when we looked at their blood metabolism, we were actually able to link climate variations to their blood because we had one year specifically that had really bad sea ice. It was really, really warm. All the sea ice melted, which disrupted the food web of these seals. And what we measured in their blood was some clear signs of malnutrition, a lot a drop in essential fatty acids, a hike in saturated fatty acids, and just some emaciated seals in general. We know that their blubber was really thin that year. And so we were able just to use a little, just using a little blood sample to link these climate variations to disruptions inside the blood of the seals. So that was pretty cool. And so there's some of the emerging tools that we can use to help us understand all these different stressors and how they may combine to affect different tissues inside the animals. So stay tuned because today our lab, in collaboration with Chloe Kotick, for example, but also our colleagues, Claire Anvik and Eve Jourdain from Norway, our colleague Paul Tixier from France, and a bunch of other colleagues were leading the largest study on killer whale health ever using metabolomics and steroid hormones, which will look at the effect of these contaminants throughout many populations across the world in the North Pacific, North Atlantic, and Southern Ocean. So we're hoping that we can prove that these chemicals are affecting our killer whales and that we can help with their conservation later. So before I stop talking, because I've been talking for a while, I know that, and trust me, I know that finding out about the effect of contaminants in is never fun. So I really want to acknowledge that. And I do recognize that it's not always fun to come listen to me talk because I'm usually not telling you great news. And there is lots of work to do as a world society to really eradicate this issue. Um, for example, when the Stockholm Convention was came into effect in 2001, well, was signed in 2001, came into effect in 2004. The main two goals were 2025 and 2028, which were to fully ban and eradicate PCBs. And based on the levels that we currently measure in marine mammals, that is clearly a failure. So we failed our main objectives that we fixed 24 years ago. And we're about to miss the 2028 target as well. We know that some of these legacies, so the dirty dozen chemicals, are still impacting our ecosystems. We also know that there's a lot of it stored in warehouses in the form of waste, contaminated waste. And we know that not much is being done to get rid of this waste. It's sort of sitting in warehouses, and anything could happen to the warehouse, and the contaminants could leach back into the environment. We also know that we keep replacing chemicals by other chemicals that have not been tested. And so that can create some new concerns for marine mammals, for our oceans, and for human beings. So there are solutions, but they will require international collaboration, resource mobilization, because a lot of countries need money to help get rid of their contaminants, and public involvement, of course. And these days, it can feel a little sad to think about all that in the current geopolitical climate, but there is a way out of this. We just need to all work together. But if you're wondering what you can do at the human level, there are a few things, a few actions that you can do in your everyday life to help with this issue. And the first one at the top is that I put is to vary your seafood choices and support sustainable fisheries to help ease the burden on harvested species. So we know that if animals start to, and especially marine mammals, if they start to get hungry, they will mobilize their fats, and with it will come the contaminants inside the bloodstream and the body. So if the whales are nice and plump and well fed, the risk is lower to their health. So thinking about sustainable fisheries is really good. Avoiding, of course, harmful pesticides, herbicides around your garden, and disposing of your chemicals safely is something that you can do every day, and that will have an impact because it will, if everyone stops using harmful chemicals in their home and disposing of their chemical waste in a bad way, it will stop leaching into the environment, stop getting into our waterways and stop polluting our oceans. So that's a good step that you can implement at home. Choosing safer furniture, textiles, and household products can also help. Look for example, jackets that do not have any PFAS or PFOS. In the state of California, for example, they're trying to ban PFOS, but not all the states in America have banned it. And in Canada, you can look for businesses that do not use any of these PFAS compounds. And so same if you can wash synthetic clothes less often at lower temperatures, it sort of helps with the degradation of the particles release from the garments into the waterways. And then, of course, finally, spread the word and support stronger laws against pollution and stronger laws for conservation of our marine mammals in BC and elsewhere in Canada. And if you're watching from outside Canada, I hope that you can vote for someone who really believes in the future of your environment. So that's it for me. Thank you so much for your attention. I would like to thank all the founders. This slide is not even accurate because there's a lot more collaborators that I need to add. I will point out that Chloe and my other colleague Ev and another friend and colleague and I founded this NGO called CTOX, which is for cetacean ecotoxicology. Keep an eye out. We just launched it, but it will look at this. The main project on it will be that big worldwide study on the effect of chemicals in killer whales, but also in the future other cetaceans. So we're really committed to help protect our marine mammals. And so thank you, because by being here and by caring, you're making our job easier. Thanks so much. And I'll take any questions.
Yes, so thank you so much for your amazing talk. If anyone has any
Thank you.
questions, please either type them in the chat, or if you would like to speak, you can use the raise hand function and we'll unmute you. We do have one question here in the Q&A, just asking a little bit more, what explains the higher level of contaminants in the Western Atlantic whales?
Yeah, that's an excellent question. Thanks, Rosemary. Hi, Rosemary. Um, the higher contaminants in the West North Atlantic are from the diet. Basically, these animals are eating higher contaminated prey because they're, for example, pinnipeds, uh, other whales, and so they have more contaminants than the fish that the whales feed upon in the east side of the North Atlantic. And so by feeding on this more contaminated prey, the whales accumulate more contaminants. So for example, if you feed on a beluga or narwhal, you're going to get a lot more than if you feed on a herring. And so that is why these whales are more contaminated.
Wonderful. Thank you. Yeah, if anyone else has any other questions, I have a question I always love to ask
Yeah.
researchers, and that is, what was the most surprising thing that you discovered during your research?
Um, Well, honestly, probably the North Atlantic killer whales were so contaminated. The West North Atlantic killer whales were that contaminated. Um, I had to go back and check the data a few times because I thought it was wrong when I saw just how contaminated they were. Um, it made no sense to me because I was like, these are a population that live in the Canadian Arctic where no one else live. I mean, no, not no one else, but very few people live in comparison to other places like Norway, for example. And I was extremely surprised. That was, I think, the most surprising thing to me.
Thank you.
Yeah.
Another question here. Um, when you're collecting the like skin and blubber samples with the darts, I'm super curious. How does that work? Like, do they just fall off and float and then you scoop them up? Or, yeah.
So the way that they're designed, there's a few different designs out there, and depending on the country and what design you've adopted as a research program, but the way that they're typically designed is that they really look like a dart. The tip of it is sort of a metal tube that will come and take like a nice little skin core into the animal. And inside they have two tiny little hooks that will allow the biopsy to get stuck in the little tube. And then they're attached with some foam at the end of the dart that will float to the surface. So as soon as it falls from the animal, you can come and collect it with a net and not fall in the water. But yeah, that's typically how we do it.
Great. Yeah, I was just so curious.
Yeah.
I was like,
Yeah, I
yeah.
I have some footage, but I typically don't like to show it because like it can be a little shocking if you're not used to it, just seeing a dart hit a whale. Typically, it doesn't involve a response. Like we don't see like a painful response or anything like that. They're very large animals, and we're only stealing like a tiny bit. Um, but I think sometimes they act a little surprised, like, "Hey, what is happening? Why are they shooting me?"
Yeah.
But yeah, it's yeah, that's how it works.
Awesome. Thank you so much. Yeah, do we have anyone else from our audience? Okay, Jay and Carolyn, Vorik, do you mind unmuting them for us?
Can you hear me now?
Yes.
Uh, how do you tell when you identify something that originates from a herring that this was due to the herring that the whale ate, or due to or originated from the centipede that ate the herring?
Yeah, that's a really good question. Thanks for asking. We actually run a lot of checks before we put all the prey into our model, and we make sure that the model can accurately distinguish which prey is which. So if it doesn't work, we just cannot use it. Luckily for us, because of this tiny metabolism that occurs in each animal, pinnipeds will have enough of a variation from the fish so that the model will identify pinnipeds as being pinnipeds and fish as being fish. So for us, it won't be an issue because they're different enough just because of this little metabolism transformation that occurs inside the pinnipeds. And that is why we needed to calculate the metabolism for the killer whales, because we know there's a little modification that happens. It's not big, but it's still significant. So yeah, that's how we know for sure that a seal is a seal and a fish is a fish.
That's so fascinating that that is a thing that you can just tell from those little samples. That's amazing.
I know,
That
and
makes it so cool.
I was very new to the world of fatty acids starting my PhD, and I just became so fascinated by what you can do with these fatty acids and just how much information they tell you about the ecosystem. Like you can also, and I didn't have time to show it, but you can look at something called a fatty acid niche, which is basically a geometric way to represent the variation in the fatty acids between populations. And so, based on the size of the circle representing that variation, you can infer a lot of things. You can understand if it has a large feeding range. Like, for example, it eats marine mammals plus fish, it's going to have like a big circle. But if it has a very specialized diet, it's going to have a tiny circle, so not a lot of variation. And you can infer all that from like a little bit of fat from a sample. Isn't that cool?
That's amazing. Alrighty, does anyone else have any other questions? Well, if there's no more questions, then we can wrap up here. If you do think of something, please feel free to reach out via email after the talk. Um, Thank you so much for
Thank
joining us
you.
this evening, um, for our wonderful audience, um, and for you, Anais. Just what an interesting talk, and yeah, what a great way to end our Café Scientifique um, programming for this calendar year. As you can see up on the screen now, we have the dates for our spring talks. So Please feel free, join us for more engaging conversations and new topics in the new year. And thank you again for everyone for being here tonight and have a wonderful evening.
Thank you, everyone.